And I was only here for a few days. It's great to be back. Of course, what I did 25 years ago was I bought a lovely hammer, which I then took back to England and never used. So it's great to be back. So I decided earlier on in this week that because there were rather few talks about topological knots in proteins, that what I would try and do was give an overview of the whole field, so both experimental and computational work in the field over the last 20 years. And I regretted the decision at about 6 a.m. this morning. I thought I'm never going to finish this talk, it's going to be too long. So I've missed bits out. I am an experimentalist, as you will see, I'm in the chemistry department. And so it's very interesting for me to come to physics and maths talks and listen, possibly not understand, the different way in which different disciplines look at the problem. Okay, so I'm going to try and cover 4 things. So to begin with, I'm just going to simply summarize what protein knots, what knots are in proteins, a few examples of where they are in proteins. And I'm going to talk a little bit about how they get there. So how does a polypeptide chain that's maybe initially unknotted fold and become knotted? And then I'm going to talk about the most controversial bit, which is why are there knots in proteins? And this is something that we still like to argue about. And then right at the very end, I'm just going to talk about some very recent work that my group's done. Okay, so it doesn't work. Sorry, this worked earlier. The laptop like press arrows. No, we click the zoom X. Can you the zoom X that's come on the screen? No. It's not look, it's actually not showing on that screen. So go out of the presentation. Go out of the presentation. There's an expression in English which this was all working fine earlier. Now is that me or you? Okay, right. Yeah, there's an expression in English which is, if it ain't broke, don't fix it. And it was working earlier, and then they made some more suggestions on what to do about it. No, don't touch it. Anyway, we are back. Okay, so I'm going to give a brief history of knots in proteins. So again, talking about what knots have been found where, how they fold, why they might be there, and then a bit of new work from my lab. Okay. Please just. Oh, okay. So a brief history. So somebody said, why did people in the protein structure, protein folding world think that knots? I don't have time to go into the reasons, but essentially there are 3 papers starting in 1974. And which speculate on whether there would be a knot in a polypeptide, folding polypeptide chain. And people put these sentences into papers and without without any further arguments. They just said, of course, we wouldn't expect anything to become knotted during spontaneous folding. Yeah, presumably a knot is not impossible if it's a very shallow knot, but it would be exceedingly difficult for a protein to fold up to knotted configuration. And then 1994, the base again looked at the structural database. In 1994, there were 400 protein structures in the database, and they found one very, very shallow knot. So only a tiny little bit of the chain passes through a loop to form the knot. And I should say, and I'll go on to see in a second, that I'm talking about open, generally I'm talking about open systems, not closed systems. So the length, the shallow or depth of the knot is really important, i.e. the position of the knot relative to the end of the chain. And again, in 1994, this was a few years after I finished my PhD, again, protein folding mechanisms only explore unknotted conformations. I think I read this paper and completely forgot about it. It was kind of like, yes, of course. Okay. In 2000, things changed dramatically because the computational protein structure prediction person in London called Willie Taylor used some nice maths, which you all know, to generate an algorithm to detect whether there were knots in the open linear chains of proteins. And I won't go into the reasons why he did this. Great. Somebody can ask me over coffee. Essentially, you will have seen this kind of smoothing algorithm where we take a very complex structure, and whenever there's no topological crossings, we reduce it to get to a smooth, simple representation of the protein structure. And indeed, what he found in 2000 was there were lots of proteins with knots in the protein database, including quite a lot of proteins which had very deep knots. So a large amount of the chain passes through a loop to form the knot. So this was really a big surprise and got some of us in the protein folding field to start thinking about how these structures would fold. So just to bring you up to date, and to mention the work of others in the field. So I'll talk a lot about other people's work. This is just a recent plot from a very nice review by Danny Xu in Current Opinions, which just plots the number of knotted and slip-knotted structures in the protein database. So the protein database is where there are high-resolution structural information on proteins. And you can see that the number has slowly been going up, both for slip knots and knots. There's a fantastic database which was developed and was run by Johannes Sophie in Warsaw called P-Knot-Prot, which contains every single knotted and slip-knotted protein that has been discovered. And I quickly checked the numbers a few days ago, and the total knotted entries is now about 2,000. That includes slip knots. Knotted chains, about 770. So that's quite a few structures, but not that many, considering that the number of unknotted structures is of the order of over 350,000. Okay. So this slide, which is from a review I wrote with Christian a few years ago now, just summarizes just a bit more generally about what we know about knotted and entangled and linked structures in proteins. So there are quite a few different examples. So we have a couple of examples of proteins which form links, either as monomers or as oligomers. We have what are called pierced lasso structures now. where there's a covalent bond formed to form the loop, and then the chain pierces through the loop. For decades, we've known about the existence of cysteine knots. So these are knots which are formed by covalent bonds in sulfides. We have slip knots, which is shown here, and then we have this class of knotted protein. And I'm just showing you this to give you some kind of general feeling for the different structures that form. But for the rest of this talk, I'm just going to talk about these knotted structures. So I will tell you now that a lot of very clever mathematicians and physicists have worked on ensuring that these these structures which have open ends are truly knotted when you connect the ends in a multiple of different ways. I'm going to tell you as an experimentalist, what we and others did was we simply pulled on one end and pulled on the other end and measured the length of the chain to show that the overall length of the chain was shorter than it should have been if it had been fully extended, to show that the knot was there. So the knots are definitely there. Again, the length of the depth of the knot, I think, is very important. You'll hear people talk about knotted proteins, but some knotted proteins will have very shallow knots. You need to take only 1 or 2 amino acid residues off the end of the chain for it to become unknotted, whereas deeply knotted structures, which are ones I'm more interested in, can have hundreds of amino acid residues. So a large part of the polypeptide chain has to pass through the loop in order to form the knot. So for 15 or more years, no, for 20 years, we had these 4 classes of knotted proteins. We had a lot of trefoil knotted proteins here, and this just shows, again, a simplified representation of a very common class, which are bacterial transferases. We knew about 4 1-figure of 8 knots, 5 2 Gordian knots, and 6 1 stevedore knot. But for 20 years, these were the 4. And it didn't look for a long time like there were going to be any more until relatively recently. My group has worked extensively on this group, which are deeply knotted, and this group, which although they have a more complex knot, it's relatively shallow. And that's why I just wanted to highlight those structures in a little more detail. Okay, oops, sorry. So this is the knotted trefoil methyl transferase. So I think you can hopefully see that it's not easy to see the knot here, which is why for 20 years they went unnoticed in the protein database. So this is the knot, it's actually a dimer, a homodimer, but that doesn't matter. And here you can see the knotting loop in yellow. And we know from the experimental and computational work now that this protein folds by the C terminus threading through the loop to form the knot. And this is quite a deep knot. So we have made constructs of this protein where there are up to 150 amino acid residues coming through. In comparison, this is the Gordian knot that we have studied. This, to begin with, I had a lot of people who said, oh, they're just odd structures. They're very eclectic. Eclectic was the word that was used. This is the 5-2 Gordian knotted protein. This is a protein called ubiquitin C-terminal hydrolase. You don't need to know what it is, but you might want to know that 5% of the soluble protein in your neurons is this knotted protein, and its levels in cells are upregulated in various cancers. Okay, so I'm going to kind of zoom forward now to the last few years So some of you will be aware that in 2021, a paper came out in Nature using AI machine learning techniques to predict the structure of proteins. It's called AlphaFold. There have been several different versions. The second version is much better than the first version, and I'm not going to talk through how it works. But essentially, it is based on the fact that now in this protein structural database, there are now hundreds of thousands of structures. And in addition to those hundreds of thousands of structures, we have millions and millions of sequences of related proteins. So it basically uses these two big resources. The bit that happened, I understand the input and I understand the output. The bit in between I don't understand. And this got more press than anything else. And I got lots of emails saying, oh, Sophie, are you out of the job now? Anyway, so what happened was they deposited all the structures predicted by AlphaFold, and you can go and look at them, and you can go and search for knots in them. And several groups did this, and they came up with some very surprising results. So there are two papers published in 2022, 2023, and in these they find AlphaFold predictions of knots which are more complex than the four families of knots that we had seen to date. And that includes, so this is a paper by Peter Vernau and Todd Yates, and they observed both 5-1 topology of knots, which we haven't observed before, and also 7-1. And they also predicted composite knots, so chains where there was more than one knot in the chain. And this shows the example of this kind of tandem trefoil knotted protein, which is predicted from AlphaFold. Joanna Solkowska's group at roughly the same time also analysed the AlphaFold structures, and in her case she used a slightly different criteria of the structures that she looked at, and she found this human protein called from von Willebrand factor, which has again a much more complex knot than we have yet to observe. So this is a predicted structure, and this is the 6-3 knot. Okay, so they're predicted structures. AlphaFold got a huge amount of hype, more hype than I've seen any science getting in the last 30 years. Indeed, it turns out that some of the structures predicted by AlphaFold are correct. So maybe I am out of the job, I don't know. Anyway, in very recent work, so I literally found this paper on Monday, whilst I was here. This is a paper that's just come out by Danny Xu's group in Taipei, in Taiwan, and it takes a relatively small protein from bacterium, which is predicted to have a 7-1 knotted structure, and they have crystallized it, and they have shown that indeed it adopts 7-1 knotted structure. So it is remarkable. So now we are really expanding the kind of universe of knotted proteins. In addition to this, Joanna Solkowska's group took the predicted tandem trefoil knotted protein and they crystallized it, and they haven't published it in paper form yet, but it's in the protein database. This is its accession number, and it is a tandem 3-way knotted methyl transferase. So the methyl transferase, as I showed in an earlier slide, it has two of these stuck together. Okay. So I'll come back to AlphaFold at the end of the talk because it doesn't always get it right. So just be a little bit careful. So now I'm going to try and, in about 4 slides, tell you about what we know about how multi-proteins fold from experiment and computation. So, okay, so this is the diagram that lots of protein folding people use, which was developed by some physicists, Sanuchik and Wolinas, who you will know, which shows a protein folding funnel where the protein in its unfolded, unstructured state starts somewhere up here on the rim of the funnel. And it folds down into, and this is a highly entropic state, I should say, and it folds down into a native state, which is got very reduced entropy, and that this folding funnel is quite smooth. Okay. And this applies to many proteins, and there may be more than one way down the funnel to the native state. And this funnel, although certainly applies as quite a good description of the folding of small proteins with simple topologies. Okay. So if people say they've solved the protein folding problem, what they mean is one of 2 things. They've solved the protein structure prediction problem, AlphaFold, which was always a different problem, or they've solved the mechanism by which small, tiny little proteins fold. Anyway, so there's also a much rougher energy landscape, and this energy landscape again starts unfolded at the top, and we go down and we can see there are various barriers and there are various intermediate states, some of which can act as topological traps. And then, the protein the chain has to get out from this partially structured intermediate state. We have to partially unfold and backtrack before it can then go down into the correct native structure. And very briefly, I will tell you that knotted proteins show experimentally, some show features of this type of knotted funnel, and some show features of this type of protein folding. And the idea that they all fold slowly and get stuck, and have to avoid topological traps just isn't the case. So again, I'm summarizing, I don't expect anyone to understand this slide, but this is just to remind you, basically to say that experimentally we see a huge variation in behaviors. So we see some knotted proteins, particularly shallow ones, they fold very fast. So their folding half time is in the order of 64 microseconds. They don't get stuck. They don't fall into intermediate states. Whereas other knotted proteins, particularly deeply knotted proteins, fold with much more complex pathways. They fold much more slowly, so many orders of magnitude more slowly. And there's evidence of multiple paths to the native state. And one of the issues in trying to summarize anything about this field really is this point, which is knotted proteins do not all behave the same. If they did, I think we would understand a lot more, but they don't. And they show a diversity of behaviors, which is very typical of unknotted proteins. Okay, so the experimental work, and I should also say, I like the idea of using the word, there's no surgery involved in the folding of these or unfolding and untying of these proteins. The chain doesn't break, the chain doesn't reform. So in some ways that's quite different to DNA. So we've learned a lot more details from the computational side, and that has included a number of mechanisms about how they fold. And I had to just kind of slightly randomly choose a few examples. So this is an example by Joanna Costa, who's done a lot of work in this field as a postdoc with Jose Panucci, now in her own group. And essentially what happens is a loop forms in a twisted conformation, which you can see here, and then there is some threading event. And this might be a direct threading event, as kind of shown here, or it might go via a slip knot. So, and they have shown that, again, this energy landscape, this protein folding funnel, can be quite complex, even though we don't always see it experimentally. And again, you can go via what's called a plug, so just the end of the chain comes through the loop to form the knot, or you can go via a slip knot, where the slip knot forms and then it's pulled through. And you may get into some kind of topological tracks where the protein, it forms quite a knotted native-like structure, but isn't knotted. And I'll come back to that again at the end. Okay, so we have these different mechanisms. We also have, so I had to do Christian's work, of course, no, but also what you see in some of the simulations is interesting because a single chain doesn't always fold the same way. If you run multiple trajectories, you sometimes see a direct threading with this blue part of the chain coming through this loop here. Again, you see some slip knotting events. And you also see what's become known as the mousetrap mechanism, where the loop, shown here in kind of green and yellow, slips back over the chain. And the other thing I wanted to just say from the computational side that we've learned is, although you can fold knotted proteins using what are called Go-like models, which only use native-like interactions, there certainly is a role for non-native interactions in the folding of these proteins. And this is work that was initially shown by Eugene Shaknovich. Again, Christian's done a lot of work in this area of non-native interactions, and Patricia Feska in Lisbon. And essentially, you just look at the green and red. This is knotting probability here. In green, you can see the knotting probability for the folding of a particular knotted protein in the absence of non-native interactions, and in red, in the presence of non-native interactions. So you can really see how, in some cases, non-native interactions may really play a dominant role in the successful folding of these proteins. Okay, so now I'm going to get, now I'm going to get a bit more complex. Lots of people talk about complexity, and complexity means different things to different people. And to me, complexity is all about what happens when you go from taking your single, your highly purified protein chain in a test tube, and you think about what's going on in the cell. So this is just a view of what it might look like inside a cell. And there's a couple of points to make, one of which it is a very, very crowded environment, which generally is not good for folding. Here you can see a ribosome, which is the cellular machine that makes polypeptide chains. Here you can see a newly synthesized polypeptide chain emerging from the ribosome. So in addition to the fact that proteins are synthesized and then fold, there are also a huge class of other proteins called molecular chaperones, one of which is shown here in green, which aid in the folding and maturation of some proteins in the cell. Not all. And this is just a snapshot of just a few of the molecular chaperones that exist in vivo. And so we have chaperones in this class, DNA, DNA, which come on and they can bind to the nascent polypeptide chain as it is being synthesized. Then this chain can be released from that set of molecular chaperones. It can be passed on to another set of chaperones, such as ROEL, ROES. Some of these chaperones are very similar in prokaryotes and eukaryotes. In eukaryotes, of course, there are a whole host more chaperones doing different things. And I think the thing that we now know from two studies is in vivo, there are aspects of the folding of multi-proteins which are almost certainly different than in vitro or in silico. So I'm just going to tell you the punchline. So this is some work my group did quite a few years ago now. That essentially we looked at the folding after transcription, transcription and translation. And what we showed, whereas folding in the absence of any molecular chaperones was very slow, these are not to be transferases. In the presence of the molecular chaperone ROEL or ROES, it actually accelerated it by orders of magnitude. So again, multi-proteins may be slow to fold in vitro or in silico, but may not be slow to fold in vivo. So in addition, from a computational study, there came out another potential mechanism for the folding of proteins in vivo, and that is a ribosome base. So as the protein chain is synthesized and it emerges from the tunnel in the ribosome, then it forms a loop by some of the side chains interacting back on the ribosome. And then as this loop is in place, then the knotted structure is formed by the rest of the chain, which you can just see here in red, pushing through that loop. So this is an example of what we would call co-translational folding and co-translational knotting, rather than post-translational folding. And I will tell you that it's very likely that this is a predictive mechanism. It's not been shown experimentally yet. The chaperones that we studied act post-translationally. So it may be that some knotted proteins fold co-translationally and other knotted proteins fold post-translationally. It would be so much easier to give this talk if they all did so, but they don't. So why? So why is the most difficult question to answer? So in the early days, it was that there are two facts, one of which is it has been shown by numerous groups that knotted protein structures are conserved evolutionary. I didn't talk about the evolutionary pathways by which knotted proteins may have evolved from unknotted ones. It's very interesting. I just don't have time to talk about it. And from the early studies, we knew that some knotted proteins folded very slowly, and the computational analysis showed that the presence of the knot slowed the folding, and we've shown experimentally that it can slow the folding of the protein for a number of systems. However, there's all that molecular chaperone machinery that might speed up, and not all knotted proteins fold slowly. So for many years, it was speculated that knotted proteins have some advantageous property over unknotted ones, which means that they have been conserved. And there was a huge focus on stability, and this came out of lots of computational studies. And there are different aspects of stability. Of stability, and I'm going to quickly go through a lot of them. So that there's thermodynamic, kinetic. So we can mechanically unfold proteins, and there's also unfolding through cellular machinery called the degradation machinery. And in order to get the last part of the talk in, I'm actually going to, and I've kind of published my views on this. I'm going to skip through all of these. Okay. My view on, so we come back to this point, and the point is some knotted proteins are very unstable. Some knotted proteins are very stable. There are 7 orders of magnitude difference in folding rates, in stabilities, in unfolding rates, in degradation rates. There's huge variety of different forces needed to unfold knotted proteins. So making a single statement that knotted proteins exist because they are very stable, they are a stabilized structure relative to an unknotted protein, I think personally is incorrect. And it may be that some knotted proteins have been conserved because they have additional stability. But coming from a protein structure world, I can give you 101 simpler ways to engineer stability into a protein other than to knot it. So I just want, that's my take-home message, is there's a huge diversity of properties. And what you can say about one knotted protein, you can't say about any of the others. Okay. I will say there's very nice work by Johannes Costa on the spout family of knotted methyltransferases that shows that the knotted structures catalyze the methyltransferase reaction differently because of their structures. Their ligand binding is different, and hence their mechanism of action is slightly different to unknotted methyltransferases. And again, I just don't have time to discuss that. So I wanted to end in the last 5 minutes, really, just to come back to recent work in my group. And there's 3 very quick projects I want you to introduce to. So Joanna's group crystallized this tandem 3131 knotted protein predicted by AlphaFold and showed that it existed. We've actually taken a different approach to make a tandem 3131 knotted protein, which is shown here. So this is not the same one. This is the different one. And we have extensively looked at its folding pathway. And I don't expect anyone to understand this slide. But I'm an experimentalist. We need data. If we don't have data, I don't say anything. So this is just to say the folding pathway is complex. And what you won't know, but I'll tell you, is this slope here. So for most proteins, this part of the kinetic plot should go in this direction, but it doesn't in this case. It goes in this direction. And what this tells us, so this is a knotted protein which folds efficiently. It gets to its native knotted state in vitro in the absence of molecular chaperones, but it's slow. But for me, again, coming from the protein folding world, what's so neat about this is, and it goes against a lot of the general thoughts that proteins just slide down this smooth funnel, that this protein gets itself into a very deep kinetic trap. It can get out of that trap, but it takes some time. And for me, I think that's neat, that somehow it can get into this intermediate state that is almost certainly not knotted or not fully knotted. But it also can, if you just leave it spontaneously, it will come out of that trap and find its way to that thermodynamic minimum. Okay, so the next one, the next project. So For those of you who don't know, which I think is the majority of people, protein structure, protein design, in the last 20 years, there has been a phenomenal amount of work done on the design of proteins, unwanted proteins, with specific structures and specific functions. Just, I can't even begin to describe the amount. In contrast, we have got almost nowhere trying to design knotted protein structures. So there's one case by Todd Yates where he used an evolutionary pathway and he basically took a homodimer, which was highly entangled, and he connected the end of one of the monomers to the start of the other monomer, and then got a knotted protein. But apart from that, there's basically been almost no work at all. So we were very lucky a few years ago to start to work with the Bradley and Stoddard groups in Seattle, and they are protein design people. And they have been using a relatively simple approach, which is where you take a kind of highly regular structural motif, kind of based on alpha helices, and repeat it to design very stable protein structures. They then went one step further, and they thought, well, if we tweak this design, if we make the helices a little longer, then we should be able to get to a trefoil knotted structure. And that's when they made this, and they showed it was a trefoil knotted structure. They got in touch with us, and we looked at its folding. This trefoil knotted structure folds very fast. And then I said to them, that's cool, but how about making a 5-1 knotted protein? Because we don't see any of those in nature. And they said, sure, we'll have a go. So then they designed, so it says 2-5, it's a bit, this is their nomenclature, not mine. This is a 5-1 knotted structure. So they also designed a 5-1 knotted structure. So the 3-1 knotted design, perfect fit to AlphaFold prediction, very good, perfect fit to experimental data. So the first de novo design knotted structure. Not confirmed variety of ways. I'm not going to go and talk about the folding kinetics of the pathway or anything like that. But the 5-1 knotted structure, completely different story. 5-1 knotted structure designed, 5-1 knotted structure predicted by AlphaFold, crystal structure solved, not a 5-1 knotted structure. This forms a 3-1 knotted structure. So for me, this is a slightly cautionary tale about relying entirely on AlphaFold predictions. And secondly, it raises lots of questions in my mind about why AlphaFold doesn't get this correct and why the design doesn't work. And we've yet to prove this, but AlphaFold and the design process rely entirely on making an energetically favorable end state, the native state. They don't in any way consider the pathway by which the polypeptide chain has to fold and get to that structure. So it's possible in this case that although the 5-1 is thermodynamically stable, it's not very kinetically. We just have to do more work to show that. Okay, in the last minute, can I have 1 minute? Because there was a bit of disruption at the beginning. Thank you, Chair. Always be on good terms. Yeah, this is just it actually folds very, very fast. Ridiculously fast, so fast we can almost not follow it. Just going to end, so on Monday, Christian gave a lovely talk about translocating knotted DNA through a nanopore. So I'm just going to talk about some recently published work where we've pulled knotted proteins through nanopores. And I was going to start with this, and this is somewhere where computationally there's been a lot of work looking at the effect of the size of the pore, the type of the knot, the strength of the force, whether the pulling is intermittent or not. But there's been almost no experimental studies on this. So Christian described kind of nanopores. So here, this shows a nanopore, our knotted protein, and we can change the size of the nanopore and under different conditions. So it can be too small to allow the native structured protein to go through, or it can be larger, in which case the protein just comes through. But essentially, we can make it small, sufficiently small that the protein has to at least partially unfold in order to come through the pore. And then you just apply an electric field and you measure the electric field. And this is a lot of complicated data. And essentially, again, we see different things with different knotted proteins, and these are different knotted proteins that are in exactly the same class of knot. They're in the same family, but they have different thermodynamic stability. And what we can see is that for smaller pores, both proteins have to unfold through several intermediate states and even get to the unfolded state. So access to these intermediate and unfolded states is key in order to get translocation, and we can begin to probe in greater detail. And I didn't show you any of the detail, but there's suggestion in the data that we may be able to use this method to detect between an unfolded unknotted chain and an unfolded knotted chain, which experimentally it's actually very difficult to do. I know it's trivial kind of mathematically, but experimentally it's. So I'm going to kind of finish there. I had a summary. I ran out of steam at about 8 o'clock this morning and had to go get coffee. So there are things we know and agree on, there are things we know but we don't agree on. There are lots of things we know we don't know, and therefore we don't yet have to agree or disagree. And there are all those things that we don't know that we don't know. So anybody who wants to know a little bit more, there are 3 reviews from different perspectives, which I put up here. And then I'm just going to end by finishing the people in my group and my collaborators who perform some of the work. Thank you.
Thank you.
Did I miss anything else?
Those de novo proteins involved.
Yes.
Maybe that's why AlphaFold the crop. They never trained anything close. Is that right?
So Phil Bradley and Mary Stoddard, their approach to protein design is based on the repeating helical units. But other people like David Baker and many other people have used a whole. Helical proteins tend to be easier to design, but people have designed also lots of just pure beta proteins now. I'm not aware that anybody's attempted to design an all beta protein which is knotted. So I can't say whether we just don't know about it because nobody's tried to do it yet or not.
Beautiful thought. I don't know if anybody has tried to find an association between IDRs, intrinsically the southern regions, and the presence and absence of nodes, and if that will have any implication on the protein stability. And the second question is about, in response to stress.
Can I answer the first question first? Otherwise, I'm likely to forget the first question once you've answered it. So it has been speculated so there's a large class of protein called intrinsically disordered proteins that don't fold into globular structures of the type that I showed you. And they're associated, quite a few of them, not all of them, quite a few of them associated with neurodegenerative diseases. So, and they can form quite long unstructured, they're large chains, they're unstructured. Computationally, people have shown that they can become knotted, and computationally, people have shown within the models used that occasionally they may inhibit cellular degradation. And this is work done by the late Marek Chiplak in Warsaw. I just have a word of caution because it's so, sorry, I don't want to give too many details. So it's incredibly well established in the cellular degradation world that the cellular degradation machines can take more than one chain at the same time and pull it through. So people have disulfide linked multiple chains together and shown that they can pass through the degradation machine and be degraded. So the idea, and we don't see, but for most knotted proteins, you can get relatively fast degradation. So I don't think, although it's a neat idea, I personally am not sure that biologically it's going to be in any way significant.
And the second question, very quickly, I was thinking about the during a stress signal like heat shock or any other kind of stress, like if noted protein could become unnoted or and vice versa, and perhaps have a biological function.
So no, I don't think anybody's investigated that. I think, knowing quite a bit about, I've worked quite a lot on molecular chaperones on unknotted systems. I think, again, there's going to be a big difference between the different knotted. So I think these proteins that have shallow knots, I think when they unfold and lose their secondary and tertiary structure, they untie fairly rapidly. I think with the proteins which have deep knots, I mean, my group's shown that if you take one of the deeply knotted methyltransferases and unfold it, it loses all the structure, but it takes a long time for the chain to untie. And Patricia Jennings' group in UCSD has also done some lovely work. So I think it's going to be dependent on the protein.
You have a question, Jay?
Thank you for the beautiful talk. I was wondering, what is the most complex protein found in experiments, meaning the one with more crossings? And have someone checked the correlation between the length of protein sequence and the complexity of the proteins found? You know, like
So the most complex knot is the 7-1 knot. So again, we're talking about the number of chain crossings, not necessarily depth. So I personally think that depth of the knot is also important. So that's the work. That's work I won't go back to, but that's work that's just coming out in the Journal of Biological Chemistry from Danny Shu's group in Taipei. So the other predicted 5-1 and 6-3 proteins that we do not yet know what their structures are. And let you said something about length, but I'm not.
yeah, like basically, you would expect at least in polymers, not necessarily proteins, that then the longer the polymer, the more complex the knot you could come from. So
Yeah, there's no correlation.
that
That 7-1 knot is in an 89 amino acid protein, which is very small.
Yeah.
You can get simple trefoil knots, deep ones, in proteins that are 5 times that size. So there's no correlation.
Maybe I can jump in with a question myself related to that. A relevant parameter besides the nominal complexity of loss number is the unknowing, which for 7.1 is pretty high. Also for 5.1 is more complex than the threshold. But maybe aiming for a complex twist nodes with unknowing number equal 1 might be interesting too, maybe simpler, maybe AlphaFold would give more reliable predictions.
Yeah, I mean, more and more people are beginning to become interested in designing, not, and you will know the work of Ivan Kutza, in San Sebastian in Spain. Yes, so people are trying to use, people are using different approaches, so coming from physics and using simple models of polypeptide chains, patchy polymers, to try and understand what types of sequence may result in knotted structures. So, but I mean, compared to, so there's another form of molecular knot that's made by synthetic chemists, although slightly different. I mean, in their case, they've made really complex knots. So compared to them, we're quite a long way behind, but they don't make a single chain and thread it. They stitch pieces of chain together around a template. So I think maybe it's slightly easier.
It's assembly, so it's a
Yeah.
very big. Other questions? Renzo?
Just a curiosity, I wonder if is any bold statement on the difference between geometrically tight knot or a loose knot. I would be inclined to think that I would imagine that maybe there is a kind of reason for having difference in functions between, for example, privileging the tight configuration rather than the loose configuration. By loose, I mean the knot distributed along the chain rather than confined in a small region.
I think with most of the protein structures, I mean, when you do a mechanical unfolding experiment and pull, you can make the most tightened knot by putting everything under force. And depending on the knot type, you see anywhere, so trefoil knots, they tighten to about 12, 13 amino acids in length, and you can, 5-2 Gordian knots that we looked at, they can form 2 different states, one of which is about 24 amino acids, and so that you can form those tightened knots. I mean, compared to those, yeah, you know, for me, the difficulty is knowing that the loose knot doesn't really exist in the native state of proteins, because the rest of, because it's not a dynamic knot in the native state. It's staying in place because it's held in place by the rest of the protein structure. So I find it, so I can only think of loose versus tight in terms of unfolded structures. But with unfolded structures, unless you have an IDP, you have no function. So perhaps we can talk about this more.
Okay. So we need to move to the next speaker. So let's thank Sophie.